تنسوا الاعجاب بالفيديو والاشتراك في القناة.
This is the conceptual argument against design. It was first proposed by Professor John MacDonald at the University of Delaware, and it has to do with the concept of irreducible complexity. Just about the day that my book came out in 1996, people were trying to see how they could build mousetraps by numerous successive slight modifications. That's a good thing to do. That's how you would attack an argument like mine. And for my money, the best one was put out by this fellow, Professor John MacDonald. And he wrote on his website, which is shown up there, he showed this trap, and he said, well, this Behe guy, he said, this is irreducibly complex. It has all these 5 parts. You take one away, it won't work. He says, but I can take away a part, and it'll work. I can take away the catch right here. He says, I took it away, and now I put the holding bar down here, and it's just kind of tucked into the edge of the hammer. And the idea is that if a mouse comes along, this is just precariously balanced. He nibbles the cheese, this will slam shut. If the mouse is unfortunate, maybe his tail will be hanging over here. This will catch the tail. He'll have to drag the trap around behind him. You won't be able to get food as efficiently. You won't be able to attract mates. And it would work. It might be a little less efficient, but it would work. But he continued, not only that, I can take away another part, he said. I can take away the holding bar, and it will still work. He says, look, I took away the holding bar, and now the hammer is shaped so that it's just touching the edge here, and the tension is pushing it down. Again, the idea is the mouse comes along, nibbles here, this goes over the edge, this snaps down and catches a paw or a tail, and the mouse is in trouble. But he said, I can build a trap with 2 parts. I can take away the hammer. Now the spring itself is shaped so that it hangs precariously by the edge here, and the same thing, if the mouse came along and nibbled, it might get trapped. But he said, yes indeed, I can build a mousetrap with 1 part. And here it is. This is the one end of the mousetrap, precariously balanced on this one, and it's under tension so that if it's released, this will go down, catch a paw or a tail here, and again, the mouse is in trouble. So he's saying that, yes, I showed a mousetrap with 5 parts. He can make one with 1 part. But nonetheless, he says something strange on his website. He says the reduced complexity mousetraps shown below are intended to point out the logical flaw in the intelligent design argument. They're not intended as an analogy of how evolution works. Well, what does that mean? You know, if they're not intended as an analogy of how evolution works, then what's the point? And what is this logical flaw in the intelligent design argument? Well, it turns out the logical flaw, as he saw it, was that I had pictured a mousetrap with 5 parts. He could make one with fewer, and therefore you can make simpler mousetraps than I had pictured. And my response to that is, no kidding. You know, you can make mousetraps any of a number of ways. My family, sometimes we use glue traps, you know. My kids have a game called Mousetrap with a lot of different parts. As a matter of fact, I had said the same thing in Darwin's Black Box, in the book. I had said we need to distinguish between a physical precursor and a conceptual precursor. The trap described above is not the only system that can immobilize a mouse. On other occasions, my family has used a glue trap. In theory, at least, one could use a box propped open with a stick that could be tripped, or one can simply shoot the mouse with a BB gun. That'll show it. However, these are not physical precursors to the standard mousetrap, since they cannot be transformed step by Darwinian step into a trap with a base, hammer, spring, catch, and holding bar. The point is, we have a trap right here that we're trying to explain. Can we get to that by numerous successive slight modifications? Well, let's ask the question, using John McDonald's intermediates, can we do that? Are they numerous successive slight modifications, precursors to a type of mousetrap that he showed as the full mousetrap? And can you get there by a Darwinian process? Well, we'll go through and show that you can't, but let's look at something else on his website that should give us pause. He said, the first step in reducing the complexity of a mousetrap is to remove the catch. Okay. The hold down bar is then bent a little so that it will catch on the end of the hammer that protrudes out from the spring. This end of the hammer might need a little filing to make the action nice and delicate. Now, what's the problem with this? Well, we've got John McDonald, intelligent agent, re-engineering the trap to make it work. But if you use your intelligence to rearrange things, that is not a Darwinian process. Okay, well, let's look then. Let's start with the simplest mousetrap, as evolution would have to do, and see if we can go from one to the other by a random, numerous successive slight modifications. Here we start out with step one, the trap here, the one-piece mousetrap. And the first thing to say is that this is the strangest looking spring I've ever seen in my whole life. I have never seen anything like that. What's more, we can tell that it was purposely designed this way in order to act as a mousetrap. If these angles had been a little more oblique or acute, if the length of this thing was bigger or less big, this trap wouldn't work. Well, okay, you got to start somewhere. So let's give them that. But let's say that if intelligence has anything to do with it in any other step, then we don't have anything really relevant to Darwin's theory. And intelligence comes along right away. For example, here we go from 1 to 2. Okay, so we have a two-piece trap now with a spring and a platform. But you'll notice this is not the spring that was here. Okay, this particular arm of it has been bent by 90 degrees, and this part shortened by a considerable length. This arm here has been bent by 180 degrees and shortened considerably. So not only do we have an extra part, but we have had to re-engineer a second part. So this is just a little cartoon to show that we can't go directly from here to here. We have to have somebody engineering things in order to get to the final configuration. But we have to ask ourselves, wait a second, the spring here was in tension on itself. Why doesn't the spring here kind of spring and relax? What's keeping it under tension? It's not hitting against another part of itself again. Well, if you look real close, you'll notice here's a little staple here, and here's another little staple over here. If it weren't for those staples, the trap wouldn't work. They're keeping it under tension. Not only that, notice that the placement of the staples is very important. If this staple was over here, that one over here, it wouldn't work. So we have not gone from a one-piece to a two-piece mousetrap. We've gone from a one-piece to a four-piece mousetrap, which has been re-engineered in the process. So where did those staples come from? Did an angel bring them from heaven? Well, no. Darwinism cannot invoke angels bringing staples for mousetraps because the angels are on our side. Okay, we go from step 3 to step 4. This is the last one I'll show you here. Now we're not only adding a piece, the holding bar here, but we also have to rearrange the hammer here in order to lengthen it and give it a couple more bends and so on. Not only that, notice that the new piece is not just a flat metal bar. It's got this little curl on it, and it's hooked around this staple here. If it wasn't for that little curl, this whole trap would be useless. So not only do we have an extra part, we have re-engineering all along the way. And step 4 to step 5 I leave as an exercise for the audience. So the point is that we'll get to that. The point is that people have talked about mousetraps. In particular, there's a man named Ken Miller. He's a professor of biology at Brown University.
Ken Miller is one of your antagonists. He is one that's written in scientific journals about TTSS, yes?
Yes.
And explain what that is very quickly.
TTSS is the type 3 secretory system. It's a subpart of the flagellum. The flagellum, I told you, has to build itself. This thing called the type 3 secretory system can pump out the parts of the flagellum out through a hollow tube where they can assemble themselves. So it can act as a protein pump, or a pump for the parts of the flagellum.
And what he's really getting at is that what he's saying is if we can show that this TTSS is a secretory, what was it, I'm sorry?
A pump.
It's a pump. It's a building block, okay, of a part of the complex structure that makes up the bacteria flagellum. And his point is, if we see this in other cells or other mechanics within cells, and it's pushing itself forward because TTSS appears in other type cells, then there's evidence that maybe it could have evolved to this place.
Yes.
Now, I want you to respond to that, because I know that sometimes a lot of the folks that are your antagonists get to write and put their opinions in journals, and when you try and respond, your articles are...
They're deemed uninteresting.
Yes, or rejected.
Yeah, they're
which gets to a question I want to ask you. The disclaimer you gave at the beginning, was that your idea, or do powers that be suggest strongly that you give it?
Well, a little bit of both. Powers that be thought it would be a good idea, and I agreed with them, because you have to realize that in the scientific community, I don't get as many laughs as I get here. And my colleagues were getting static because of the crazy guy down the hall from them. And they were getting emails from other people asking if Lehigh University was turning itself into a Bible college, and graduate students from Lehigh who were going out for postdoctoral interviews were being laughed at because I am at Lehigh.
You're on the faculty.
I'm on the faculty there. So
and this is a lot of what the movie Expelled really is about. It's about some of the pressures within the scientific community to, I think what they say is the science of intelligent design is neither intelligent nor science.
Yeah, they do that. They say very strongly, but they don't have any more reason for it than I've been showing you with some of the responses to my
exactly.
To my
so it gets to an ad hominem or to the man. They make these statements. They call it science in a cheap tuxedo, and yet when the evidence for that claim are brought forward, they don't stand well on the stage up against it.
No, that's correct. I'm trying to show the best responses to my arguments, and as you can see, they're just not that impressive. But nonetheless, somebody will bang a gavel, as the judge says, and said, "No, you're clearly." You're clearly not scientific. You go away. It's more or less like people in the Soviet Union a long time ago who objected to communism. They were put in mental institutions because if you didn't think communism was the right system, clearly you were not functioning very well. It's very similar to that.
Or you were exiled to a very cold, lonely place, which is a little bit of what's happening. I want to come back this a little bit. I want to get Steve back up here to talk about the evidence for intelligent design within the digital code. One last question before we move. How do I get a subscription to that Cell magazine? It just looked absolutely riveting.
Just write to MIT Press.
Okay, thank you. So that was flooding in on our text line, and I wanted to go ahead and get to that. If I could. All right, let's yield to Steve, and then we'll come back up, and we might look at Mr. Miller's tie clip. Or do you want to give a quick explanation of what that slide was, since they got a peek at it, and will Steve come out here and be ready to go as soon as you're done?
Kenneth Miller says, well, I can take away a piece and it won't function.
Steve, talk about the evidence for design inside the code, and why don't you do this? Build this bridge, because when most of us laymen think of a black box, we think of that which is indestructible inside a plane. And that's not the black box that Mike was talking about. He was talking about Darwin could not see what was inside the cell. He believed that, in fact, if the cell was as complex as it was today, he may never have written the book. He would have surrendered at that point and saying, there is such genius inside of that. It, in fact, violates what he wrote in that one quote that you had from Origin of Species. So let's go further now, not just in the complex machinery that Mike talked about. Let's talk about even a step beyond, which is the digital code embedded in that. There's a segue.
That's necessary to build it.
There we go.
Absolutely. And hey, Mike, we're not used to getting that kind of a reaction. How'd you warm them up like that? All right, I'm here to talk about the digital code. I hope we get to talk a little bit more about that type 3 secretory thing later, because I've been working with one of Mike's colleagues who's a microbiologist who's been defending Mike's honor, because there's a genetic story behind that objection that I think has shown Mike to be entirely correct, but maybe we can come back to that in Q&A. My job tonight is to talk about I think one of the most fascinating discoveries in the history of humankind, not just science, but it is the discovery that Watson and Crick made of the digital code in DNA. The first thing they discovered in 1953 was the beautiful double helix structure of DNA. We all know this structure because it's a cultural icon now. We know that DNA, you convict criminals with their DNA. We see DNA on all kinds of things. The double helix is an icon. But it's a beautiful structure. But more important than the structure that Watson and Crick discovered is the mystery that they solved and the mystery that they created. First line of the book that I've just completed is this. It's when Watson and Crick discovered elucidated the structure of the DNA molecule, they solved one mystery, but they created another. The mystery that they solved was the mystery of where hereditary information arises. Since ancient times, people have known that like begets like, that children are like their parents, that organisms are able to copy and replicate themselves in very similar form. And scientists have known that there must be some kind of signal, some sort of memory or that allows organisms to reproduce themselves. But where does that memory, that information, that signal reside? When Watson and Crick discovered the structure of DNA, there was a hint in the structure that suggested that the DNA not only has this beautiful helix, but it is embedding some kind of information down its spine. It was suggested by Watson and Crick in their very first paper that this might be the case. But in 1957, 4 years later, Francis Crick, having taken stock of all the developments that were going on, not only in the DNA chemistry world, but also in the world of protein chemistry, put forward what I think is really the basis of modern biology, which is the sequence hypothesis. And in it, he said that there are 4 chemical characters along the spine of the DNA, they're called bases or nucleotide bases, that function just like alphabetic letters in a written text or digital characters in a piece of machine code or software. This is known as the sequence hypothesis. According to Crick, it's not the chemical properties of these bases that is important. Rather, it's their arrangement. Just as it's not important what the weight or the composition of these Scrabble letters is. What's important is the way they're arranged to spell out a message. It's the sequence of the arrangement that it determines the function, not whether the letters are put on wood or tile or plastic. This was the sequence hypothesis. And here's a picture of the DNA molecule. You see the double helix shape. That's made of the sugar phosphate backbone on the outside. That's the medium of the genetic text, the genetic information. But the information is stored on the inside of the molecule in virtue of the specific arrangement of these particular characters. We represent them with A's, C's, G's, and T's. There are 4 different chemicals, and it's the arrangement of those chemicals, again, that determines the function of the molecule. Now, that's all well and good, but what exactly does DNA do? And Watson and Crick said that the characters function like alphabetic letters in a written text or like digital characters in a machine code, that's great, but what does the machine code do? What do those instructions tell the cell to do? Well, right as Watson and Crick were elucidating the structure and the function of DNA, well, the structure of DNA, there was a parallel development in molecular biology in the area of protein chemistry. It started in about the same time, 1952. A scientist named Fred Sanger was discovering about the protein insulin. Other scientists began to discover the three-dimensional structure of proteins, and that they discovered that they were immensely complex three-dimensional shapes. It defied everybody's expectation. They thought proteins were going to be simple things like hat boxes stacked up on top of each other. The ultimate secret of life, after all, should be law-like and orderly. But proteins weren't like that. We know that proteins do, and they knew that proteins did lots of important jobs in the cell. You can think of proteins as like the toolbox of the cell. Inside the cell, or in a toolbox, you've got a hammer, a wrench, a saw, you've got a plane. All these things perform functions, and they perform the specific functions they do in part because of the specific shapes they have. The same thing is true of proteins. They have very irregular, complicated shapes, and in virtue of those shapes, the proteins are able to do specific jobs in the cell. Here's just a simple illustration. You have a protein that acts as an enzyme. It catalyzes a reaction. In this case, a reaction that breaks apart two sugar molecules. But the ability to break apart that disaccharide depends on a very precise three-dimensional fit between these two molecules. The disaccharide nestles into those two grooves in this particular enzyme. And everything in biology works according to that same kind of specificity of fit. Proteins make up the structural parts of those nanomachines that Mike Behe was talking about. All those parts in the bacterial flagellar motor and other nanomachines are made of proteins. They fit together and form the machine because of very precise hand-and-glove fit between the parts. Same thing is true for enzymes catalyzing reactions. Same thing is true for proteins that help process the information in the cell. All proteins function because of this very precise three-dimensional specificity. How do they acquire that specificity? Well, we now know it has a hand-in-glove fit, another take on specificity. We now know that proteins acquire that specificity because of the way their individual subunits are arranged. Now, the subunits of proteins, that sounds like a big scientific word, but hey, I've got snap-lock beads tonight. Ages 2 to 4, it says on the box. Okay. All my best visual aids are stolen from small children, you know. Okay. Here's a protein is a long chain-like molecule that folds into a very specific shape. And it might fold differently depending on the arrangement of the subunits, the amino acids. So each of these colored beads with a different shape represents a different amino acid. And you can see that there's a lot of different ways that we could arrange even just these few beads. Depending upon the arrangement, the shape will be different. It will fold in the protein, or the chain will fold into either a functional protein, or it may lie limp and do nothing at all if the arrangement isn't correct. So the amino acids, and there'll be a test on this particular, these equations afterwards. The amino acids are sequenced in very particular ways so that the protein folds in the right way. Get the sequencing wrong, you don't get a functional protein. So Francis Crick, in fact, said that he likened the amino acids to those old typeface blocks that are used in the old newspaper headlines. They all have the same basic chemical structure, but there's a different sticky outie with each one, a different font. And the arrangement of those sticky outies determines the, more technical terms, sorry, and it determines the folding and therefore the function. Okay, so we like to say that amino acids have a property, or proteins have a property called sequence specificity. If something has sequence specificity, the function of the whole is determined by the specific arrangement of the parts. We find that in language, we find that in computer codes, we find it in proteins. But the question is, how do proteins acquire that specificity? And this is where Francis Crick's sequence hypothesis came in. According to Crick, the characters along the DNA spine function like alphabetic letters, But what they're spelling, what they're causing to happen is the sequencing of amino acids, which in turn cause the proteins to fold and therefore produce a functional tool in the cell's toolkit. So information on DNA is transcribed, translated, and produces the functional outcomes, the protein machines that the cell needs to survive. So you've got a form of functional information, sometimes referred to as machine code or assembly instructions in the DNA molecule. Now, this is what I call the DNA enigma. And I said at the beginning of my book that Watson and Crick solved one part of that enigma. They solved the question of where did that information come from? Where did it come from? Now, that's an important mystery for two reasons. First, because we know in our own digital world something about information and its importance. I actually used to ask my students, if you want to give your computer a new function, what do you have to give it? Software, code. Well, the same thing is true in life. If you want to build a new organism from an earlier, simpler organism, or if you want to build life in the first place from simpler chemicals, you have to arrange the parts in very particular ways to make functional organisms. To provide new biological function, you also need information. Same principle is in the software world. And that is a profound code is necessary to build new form and function in biology, just as it is in high technology. Now that raises creates a profound dilemma, because there's a question that scientists almost universally agree has not been solved by any materialistic theory of evolution. We can debate about whether Darwinian evolution is true or not, but I'm going to set that aside for tonight and look at a more fundamental issue. If you think of Darwin's great tree of life as representation of the history of life, all the things out on the branches, the giraffes and the dolphins and the birds and the fishes and us and plants, all of those things are the modern forms of life. But if you go back far enough, according to Darwin, you get to the simple organism that is the progenitor of all life. And he and other scientists have usually conceived of that as some kind of simple cell. Sometimes this is called amoeba to man evolution. That's not quite accurate, but you get the idea, okay? But the question that is raised by the recognition of the centrality of information is one, how do you make those transitions from one form to another, given that you need a lot of information, but to provide any new function, any new form? But an even more fundamental question, and which is the one I'll talk about now, is how do you get life in the first place? Life depends on information. DNA runs the show in even the simplest cell. It directs the construction of the proteins that even the simplest cells need to survive. So where did that information come from? Watson and Crick did not answer that question, but Crick in particular appreciated the gravity of the question. Now, I first came across the second question, what I call the DNA enigma, here in Dallas. I happened to be working here in the early 80s. I was right out of school. I was a geophysicist for an oil company. I learned to speak some good oil company Texan. I was looking for all out in the Gulf. Okay. Yeah. And you know how we spell all down here. It's A-H-L, all right? And so I had no inkling of this debate. I wasn't involved in this kind of thing. I was doing practical applied science. I was looking at doing digital signal processing of seismic surveys. Interesting stuff. But one day in February of 19... that's the other anniversary. February of 1985, I walked into a conference literally off the street. I'd heard about the night before at the Dallas Hilton downtown. It was a conference that had been funded by a Dallas businessman named William Garrison. If he's here tonight or in town, all I can say is you have no idea what you set in motion. And Mr. Garrison funded this conference. It was a conference of people who held a materialistic worldview and people who held a theistic worldview, but these weren't ordinary people. These were Nobel Prize winners. They were the top cosmologists, origin of life scientists, and people who did neuroscience. The three questions they were addressing were the origin of the universe, the origin of life, and the origin of human consciousness. And the conference literally changed my life. I walked in, I sat down, and there was Alan Sandage in the first session announcing how he had become a religious believer because of the evidence of the new cosmology which pointed to the beginning of the universe. He'd originally been slated to speak on the side of the materialist atheist position and had unceremoniously walked across to sit with the other guys. In the next session that afternoon, there was a discussion of the origin of life. There was some apparently young upstarts in the discussion, a local chemist named Charles Thaxton, who had written an important book called The Mystery of Life's Origin. And he was on the panel with Walter Bradley, a very established professor of thermodynamics and polymer scientist from Texas A&M. On the other side of the panel were leading origin of life researchers. But someone had joined Thaxton and Bradley that was completely unexpected as well. His name was Dean Kenyon. He had published the leading chemical evolutionary theory about the origin of life in a book called Biochemical Predestination. It was the leading college-level text on how life started from simple chemicals. At this conference, he announced publicly for the first time that he no longer believed his own theory. In fact, he explained why it wouldn't work. And he went on to express support for something he called the design hypothesis. And he said that he agreed with Thaxton and Bradley that the information that was required to build the cell pointed powerfully to a designing intelligence. Now, when he said that, well, you can't say this in a church, but something broke loose. I mean, it was unbelievable. The reaction to this was part of what intrigued me, because up to that point, everyone agreed, there was no doubt about this, that the state of origin of life biology was in a state of impasse. Everyone agreed that there was no undirected evolutionary process that could explain the origin of the complexity of the cell, and in particular, this crucial information problem. By the way, nothing has changed on that. If you saw the movie Expelled last year, you saw at the very end of the film, Richard Dawkins, who is not known for his restraint and expression of support for evolutionary theory, acknowledged that no one knows how life first originated.
لا تنسوا الاعجاب بالفيديو والاشتراك في القناة.